to hit record. um, I wanted to reiterate some of the things that are going on. Let's see, like right now and through the end of the semester. Friday, I was able to get kind of like everything up and published. So hopefully you can see it. Basically, everything is up through the end of the semester. So that's exciting. And you should be able to see it. So I do also have some muddiest points prepared for today based on some of the muddiest points that I noticed when I was going through everything this weekend. Probably saw that I was like quite active on Canvas this weekend. I'm sorry if you got a million notifications. Just wanted to catch up when I had the opportunity. And for some reason, I actually had the opportunity this weekend, which is really nice. So before I get started, I will be able to answer any, sorry, goodness gracious. Hopefully everybody's okay. I want to be sure to be able to answer a lot of your questions. But before I open it up to questions, just about anything, I wanted to go over some things over the next couple of weeks. So let's start it, shall we? Let's see. I hope you are digging my turtleneck. I don't normally wear turtlenecks, but it's cold here in Missouri. I am cold. I'm like I don't have a coat. This is brand new because I didn't know what else to wear. I don't, like I live in Arizona. I don't know what's happening. It is cold here, and I forgot that happens elsewhere. So, yeah, I heard it's supposed to cool down in Arizona, so I'm wishing that for all of you as well. Okay, so let's talk about this week going into next week. This week is pretty super light, and I hope that it is super light and that you use it to be super light, because you do definitely deserve a break before we go into the last week of classes, especially. I know several of you have a lot coming up. So I just wanted to go over what to expect. What's going to happen as for wrapping up Bio 181, and some just like what I'm going to be talking about the last couple of classes. Just going over some of the muddiest points from weeks 11 through 14. So I think that we covered a lot of the muddiest points from weeks 11, but Unit 5, I just wanted to make sure, because we've been talking a lot about wrapping up the course, but we haven't been able to discuss some of the most topics where you felt a little bit unsure. So I wanted to make sure we had time to talk about replication, chromosome terminology, mitosis and meiosis, and then some muddiest points from last week regarding inheritance. Although when I was looking through the discussion boards about inheritance, it seems like everybody felt pretty good about it. So that makes me happy. As a microbiologist, bacteriologist, inheritance is not my favorite thing. And that's actually something I struggled a lot with in when I was taking this class a long time ago. I struggled a lot because I don't know why. So it's a relief when I hear students say like, oh, I enjoy inheritance. It makes sense to me. That's great. If you're not feeling as confident about inheritance, I do want to just mention that technically that is not over. So you're not meeting for Zoom labs this week. I mean, we only have like today, so you're not meeting for Zoom labs this week, but you will be meeting for Zoom labs next week and for the last week of classes. But the purpose of those Zoom labs, you're basically just going to be working together on inheritance problems. So doing Punnett squares, analyzing Punnett squares, talking about probability, stuff like that. So that's kind of what you can anticipate. So if you're not feeling 100% confident on inheritance, that's okay. The whole purpose of having labs after Thanksgiving is to kind of get you feeling confident about it, which is good because, as I talked about, kind of where you jump in Bio 182 is basically after inheritance. So good thing to kind of like get you feeling good about it. So for this week, I think we only have classes Monday and Tuesday. And I don't know if I'm like, I thought that we had classes on Wednesday, but unless Professor Chichi is playing a prank on me, she told me that there were no classes on Wednesday. And so I will check, but even so, I mean, I can be available on Wednesday, but if I'm not planning on holding class, but if you want to meet with me or talk with me, I can be available on Wednesday. Yeah. And also we will be meeting every single day of the last week of classes, Monday, Wednesday, Friday, optional, of course, it will be recorded, but I'm just going to be going over muddiest points and having office hours and stuff like that. I have asked the program lead for forensic science. I know several of you are forensic science majors and interested in maybe in research in that field. And so I've reached out to the forensic science program lead to see if maybe she can come and talk to you about that program. So one of those classes may be dedicated to that if she can fit in her schedule, so I hope. Aside from that, please, I have your last muddiest points. It is in the week 15 module, which I'll show you in a minute. It's just called Muddiest Points Final Topics. Let me actually share that. Just going to share my screen, so hopefully you can see it. Not that. For week 15, you'll see Muddiest Point Discussion Final Topics. I just want to note that the due date on that is November 29th, which is the Sunday following this break. So it's this coming Sunday. So what I would recommend doing is filling it out either today or tomorrow so that, you know, it's not the Sunday following a break and you're like, oh, we have an assignment due. Go ahead and get that submitted early. It's open now, and that way you won't have to do it. You won't have to remember to do it after break. And also that'll help me a lot because then I can start working on muddiest points for next week, which would, you know, help me out too. So that is there. You also have a Labster that is also due the Sunday following the break. Again, I really highly recommend it as shown on the recommended schedule. I would highly recommend trying to do that Labster either today or tomorrow. I forgot to put muddiest points on here. What was I thinking? I'll have to update that. But I would try to do it on tomorrow or today, whatever you feel comfortable with. But there is a Labster this week. So make sure to get that done, but although it is not due technically until Sunday. But I'm just trying to make sure that you get a big break. Also, importantly, all of your final assessments have been published. So the Unit 5 review is open. You have 3 attempts at that as usual, and your best attempt will be the one recorded. Make sure to get those done. Those close on the Monday following this break. So November 30th, and then your Unit 5 checkpoint will open next Tuesday, so a week from tomorrow. And you'll have until December 6th to get that completed. And then your final assessment, this is the big thing, ePortfolio reflection questions. So I talked a little bit about this on Friday. So if you weren't in class on Friday, that's totally fine. The lecture from Friday is recorded. You can find it in the week 14 module. Where did I? Right here, recorded lecture from 11:20, where I go over the ePortfolio reflection questions. I do want to mention again, if you look at the recommended schedule, I have you today, like work on a couple of them. Tuesday, work on a couple of them. Saturday, Sunday, work on a couple of them, because you can save and come back to it. I would recommend, as I mentioned on Friday, trying to be sure to like copy your answers into a Word document so that you don't lose your work, even though it should, you should be able to just exit and it saves your work, but better safe than sorry. Do not do not wait until December 6th to do this assignment. Some students have completed it, and I think that on average, it took them 8 or more hours. So that is a full day. And so again, the reason I recommend doing 1 or 2 of these questions a day is that way you're not spending Sunday, December 6th, frantically trying to do these questions all in one day. It is a reflection assignment. Each of these questions take time, and we can't give an extension beyond the 6th. So technically all assignments were supposed to be due on the 4th, so the 6th is already an extension. I figured just do it instead of ask permission. But like I said, if you want to get it out of the way, it's open. It takes a lot of time. Some students have completed it. One student who I thought completed it in a very quickly, it took them 8 hours. So it takes time. Break it down if you need to. The questions aren't overly challenging. I feel like you're going to be fine having a fine time with it, but it is time consuming. So make sure you give yourself time to do that. But make sure also take a break, work on these. I have you working on them Monday, Tuesday, if you want to, a little bit over the weekend, or take a break. I mean, I think you'll be able to work on it next week a little over time if you can, but take a break. Wednesday through Friday, take a break. Your mental health is very important. And if you like pie, I linked my favorite pie recipe. So there you go. Please also don't forget that there are bonus assignments. So I discussed this assignment on Friday, the recipe bonus that is open and available to you. And also the bonus discussion, which is talking about tips for success in this class. Looking at that, there's already some really good feedback in that I think, a lot of people would benefit even from reading. No. So everything is open. So make sure to get those muddiest points done, work on, for sure, the Unit 5 review. And again, try to work on those ePortfolio reflection questions. I mean, 1 or 2 a day will help you a lot. And then also the bonus. Again, oh, hi,
sorry.
yes.
Just to clarify, we don't have a Learn Smart reading assignment this week, right? Because we're done with material.
No, thank you so much for asking that question. There is no, last week was the last Learn Smart reading assignment of the semester. There were before we got the news that the semester was going to be shortened, we were going to have content this week and next week, but we had to cut that content just because they cut the semester short. So there originally were 2 more Learn Smart assignments, but those have been deleted. I deleted them in Connect. I also thought I deleted them in Canvas, but if they're still showing up on your to-do list, I really don't know how to control that to-do list. So, okay, good. They're not there anymore. Good. So yeah, no more Learn Smart reading assignments. Again, the only content related stuff you have this week is the muddiest points and the inheritance labster. So no reading assignments. Yay. Yeah, good question. Thank you for asking. All right. So, where was I? All right. So yeah, the only thing you do have a labster just over inheritance. Again, we just want to make sure to get that solidified because it's a really important objective moving forward. Just an FYI for next week, I am planning on holding class every single day, like usual, sort of like optional attendance. It will be recorded, but if you look at the discussion board, every single person recommends for great success in Bio 181 coming to lecture. so come. Although I do realize that next week is gonna be rough. So, you know, come if you can. And then just make sure you're doing your final assignments. Labs, next week, you will meet with your Zoom lab and you'll be doing an inheritance problem set. So we'll talk about that. One more thing that I wanted to talk about before I open up to questions and move on to Muddy's points is we held a Bio 181 community circle when we had advising come and talk to you a couple of weeks ago. And it was just so nice. And we just really appreciated the feedback. That was more just sort of like talking about empathy and learning about how to communicate and talk to each other, and just sort of like making sure you knew that this class was a really, good space for you. And a lot of conversation, there's a lot going on in the world, in the, everything right now. And I'm on the management team for math and natural sciences. And so I was talking to our director and I was telling him how I thought that the listening circle we did, the kind of in class, was really great, and how I wish everybody would do that. And we also started thinking, we always want to improve. So as far as math and natural sciences, we really want to give you the best everything, right? So we really care about you. I mean, as for like, I've worked in a lot of different departments, and I will tell you that a lot of the departments that I've worked in higher education focus a lot on research, which research is very, very important, but what research doesn't always really involve teaching, effective teaching, or, working with undergraduates and, just sort of like that. And it's more just sort of like, we need to write grants, we need to get money, we need to like have these projects going, and teaching is sort of like a side thing. This is one of the first departments I've ever worked in where teaching is like a really important thing. And that sort of was emphasized with my hire, because I really focus on pedagogy. That's really kind of my background. I don't do a lot of science research anymore. All of my research primarily focuses on effective pedagogy. And so, which pedagogy is just like teaching, just like how to teach best. And so my background is in community colleges, where that is like really their focus. And so they brought me on here because they want to emphasize that. And I think that's something that's really special and unique to ASU West and even math and natural sciences. And so I'm really lucky that we've had wonderful directors. Our new director, Dr. Vernon Morris, is an atmospheric scientist. And actually I had a meeting with him this morning at 8, and I was on the meeting with him to tell you how important he is and cool he is. I was on the meeting with him, and then he's like, oh, I have to go. I'm getting a call from like the Biden-Harris campaign. And I was like, okay. That's obviously way more important than me. And so he's an amazing guy because of what he does, but he also, he and I get along very well because he has just such a great heart, which is obviously what's very important to me. And so he really, really, really cares about the students. And so we talk a lot. And I really was hoping, because we, again, want to serve you best. And so we figured what way to figure out how we can improve and how we can offer you the best experience possible. Despite the circumstances, in light of the circumstances, but even after everything goes back to normal, we just want to hear from you, your experiences and everything like that. And so we set up a very similar thing to what we had in class on Wednesday, December 2nd. And I will have a flyer for this. You saw I had Canva pulled up, so I'm going to figure out how to do that. But on Wednesday, December 2nd, from 2:30 to 5, although we both made sure our schedules are free after that as well. Dr. Morris and I will just be hanging out in a Zoom room. And we're hoping that students that are in the math and natural sciences program would just be willing to share with us just like their experiences and any challenges you have faced, again, in the circumstances or anything like that, any ideas you have. So you have experienced a lot this semester. And so how can we improve, for on-site labs next semester or moving forward? What are some ideas you would like to see? And, what are some of your anxieties this semester moving forward? We also just love to hear stories, whether that's a story of, how you pushed through or how, you needed a little more help and what that help was. Honestly, anything. So Dr. Morris is amazing at listening. I hope that I have proven to you that I am listening, but this is a judgment-free zone. It is a completely safe environment. I mean, we're pretty much gonna just like sit back and let you tell your stories. Originally, this was just gonna be me, but I thought that this would be a really good way for you to see that your input is valued so much by our department that the director of our department really wants to hear your voice. And so I thought that was really important. So I am, part of the management, but at the end of the day, like I wanted you to know that your voice goes even farther than me. And he is excited about this. This is going to be our first community circle. I know it's right in the middle of finals week, but we thought that might be a good place where you can come and share. If you're having a rough time, like come and tell us. How can we make it better? We don't know what spring 100% is going to look like yet, but let's, how can we make sure that you get what you need in the spring? So that's why it's in the middle of finals week. If we hear from you that you would like one during intersession or innovation quarter, that's great. We can offer another one. And we plan on making this part of the math and natural sciences culture. We would like to have one every month moving forward in the spring, because again, we just really, really value your voices. So consider coming. I obviously am very biased in thinking that you are just like incredible students with just wonderful voices and wonderful input. I would love to have you there. Again, Dr. Morris will be there to discuss anything or just listen or hear you. I think that's the most important thing. So come on by. If you want to talk to Dr. Morris and not have me there, come in and I can make that happen too. That's totally fine. Again, I can't speak highly enough of him. He is incredible. And I think you'll really enjoy meeting him. So again, just the opportunity to like talk with our amazing director too. It's just like, that's great. So, all right. So with that, all right, so mark your calendars, come. With that, and I will also tell you, for those interested, Professor Chichi and I are also working on a similar thing for spring, because so many we had a lot of fun just like hanging out and talking to you that one time she came to class. And I know that a lot of you were kind of thinking, oh, can we do this in the spring? And so Professor Chichi and I are working on something for the spring called just like a bio break, where we kind of have just like shared office hours that are not like formal office hours, but it's just like where we're hanging out, where we're just hanging out. And you can come and talk if you want or not, but it's just sort of just like, hey, let's hang out together. So for those who were interested in that, that will be happening. All right. And we may be arguing whether viruses are alive or not. Okay. I always talk too much about this stuff. I'm sorry. But I can move forward. I do have muddiest points. I was going to start with chromosome terminology, mitosis and meiosis. We do have some genetic mutation and prokaryotic gene regulation. But this, when I was going through the discussion boards, this seemed to be a big point of confusion. I also want to talk about replication, but that will probably be put on hold until Monday. So that's what I'm planning on talking about, because I can continue talking. However, I also value listening. So are there any questions or comments or anything about anything? Feel free to like raise your voice. Let's see. I see a comment in the chat, but it is a private chat. So I'm going to paste that and I will look into that. And so, yeah, if you, so speaking on the question, the private message I got in the chat, if you cannot go to that listening, the thing on Wednesday, that is totally fine. So I want you to know that if there is something that you want me to bring up, I like I said, I am on the management team. And so I meet with our director, our associate director, our area heads, like our program leads. I meet with them 2 to 3 times a week. At our department meetings, I give talks every week. And so your voices, based on I really appreciate the feedback that I was given in the chat right now. I see it. I just copied it into a Word document so that I can bring it where it needs to go. But please know that you can reach out to me. So that's now, that's in the future, that's next semester, that's like anytime. That's when you're like a career scientist winning the Nobel Prize. Definitely reach out to me then. You have your voice. I am, very privileged in the way that I get to hear your voice. I consider that a huge privilege. And I am very privileged that I am in a position where I can raise that to people that need to hear it. So yeah, so I hear you. I appreciate the feedback I've got in the chat. Please come if you can, but if you can't, no worries. Let's see. And I just want to take a second to read this really fast because I think, let's see, an exam and a final in a week. So I hear you. So I'm not, this was a private message. And so, but I do want to address it without giving away the anonymity. A lot of you are very stressed about the last week of classes. And I want to tell you that I hear you. So as for dropping finals week and making everything during the last week of classes, I wish that I could give you an answer for why that was done. It has been extremely frustrating because as professors, we care so much about you, doing well and having the opportunity to do well, and kind of cutting off finals week was alarming. I hope that your instructors will listen to you. So we have been pushing out the message consistently. I mean, like I send a message every Monday morning, saying like, please remember empathy for the situation. Our director sends out a message every week saying, like exhibit radical empathy. This is a situation, that's very challenging. So I think that, instructors were really thrown for a loop trying to figure out how they can meet their needs for the course, hit the learning objectives for the course. I mean, I will tell you the communication for like how we were supposed to do this was changing up until last week, which was super frustrating. It's been frustrating. And I know that has been a huge concern for you all as well, because you've probably seen your schedules change and like end of semester plans and finals, which is another reason why Professor Gillen and I were like, we got to give our students like a week and a half, like 2 weeks to like, we got to do this with the final, because we're not going to like, this is this is too much. We don't want the last week of classes. This is the first time we've ever had to worry about students having multiple finals in one day. That's not supposed to technically happen, and which is the whole purpose of finals week. So with that, I think that, please reach out to your instructors. We are given directives to, if you have more than a couple finals in a day, we are supposed to work with you. So just reach out to your instructors. To an extent, there's like only so much we can do, but we are asked to like work with you, so if you have a ton of stuff going on, reach out to your instructors. My hope is that they will be willing to work with you. And again, however, you know, I think these are really important concerns because they're concerns that we have as well. And I will tell you that when the announcement was made to cut off finals week and that we're supposed to like just wrap up all the semester in the last week of classes, so many, all of our faculty reached out in opposition. We were like, why is this happening? This is a terrible idea. Like, we just don't understand. Like, this is undue pressure. What are we supposed to do about this? And we were given some leeway. Like I keep saying, like, we have our final due on the 6th, even though technically we're not supposed to do that. It should be wrapping up on the 4th. But we were like, that's, I kind of took advantage of the fact that I have a little bit of, this much power in that situation. So it is, I hear you. These are concerns that should brought up. That email, I copied it into the Word document, and I will bring it up to our director. I do want you to know that, that is heard, and your instructors are not happy about it either. So I apologize. I don't know who to apologize on behalf. I don't know who made that decision. I apologize that was a decision. I'm not 100% sure. I see a question as for like rounding up of grades. I don't round up grades because we have flexibility built in. I don't round up. Technically, like, the grades are already round up, I guess what I'll say is. So if you look at the grading system, like an 89.95 or something is like an A-. I need to double check the grading system, but I don't round up any. My encouragement would be to, if you haven't already, do the bonus, because that can push you over any marks that you're sitting at. As for overlap between, so I see a question in the chat and it says something about having a final exam that will cut into another one of your classes. Next week is not technically a finals week, and so your classes should not extend beyond their scheduled time. So if that's happening, let me know. You are still supposed to be able to complete assignments within the scheduled class time. It's not like an extended schedule. So if you're seeing some difficulties there, like, let me know. So I see that, and I just want to make sure that's not a normal thing. Like, we're not supposed to hold you longer than you're scheduled for a typical class time. If you are having any issues with that, again, we are very flexible. I want to tell you about what's going to happen in lab next week is that you will be working on inheritance in groups. There's like 3 parts to this inheritance problem set. You'll be working on it as a group, but then your instructor is going to go through the questions with the class. And if you come to lab and go through it, you will have all of the answers. So you'll get like 100% on the inheritance problem set. Whereas if you can't come to lab, like we can technically make that an individual assignment, but you won't have the one-on-one feedback from the instructor. So we can't guarantee like a 100% on that. So I hope that clarifies things. I think coming to lab would be a really great thing to do. Obviously, if it's not possible, but my concern is with that private chat that instructors are not supposed to hold you longer than a typical class time. So reach out to me individually if that's a concern. Okay. All right. We do have about 15 more minutes. Yeah. Do you want to talk about chromosomes or do you want to talk about something else? Or do you want to just peace out? I'm going to hang on as long as you want me, but I don't want you to feel like, I know that this week, whew, I get it. So are there any questions? I want to make sure that I can answer them. Okay. No questions right now. Okay, well with that, what I'm going to do is I'm going to go ahead and go into chromosome terminology. Yeah, I agree. It's awesome to talk about. Let's talk about chromosomes. Let's talk a little bit about chromosome terminology. I know there was a lot of confusion about like sister chromatids, homologous chromosomes, homologous pairs, stuff like that. So I'm going to do my best to try to resolve that. So let's check it out. I see some questions in chat. I will get to those, but I think what I'm going to do is move on to chromosomes and then I will get back to the chat. Okay. But again, feel free to unmute, type in chat, anything. But for a little while, I'm going to talk about chromosomes for about 10 minutes. Okay, so this slide was everybody's favorite slide of the entire semester. This slide is talking about chromosome terminology, and hopefully this slide makes a little more sense now that you've gone through these lectures, like you've gone through mitosis and you've gone through meiosis and you've seen inheritance. But I just want to reiterate a couple of things. Okay, first, chromosomes are simply structures that are made of chromatin. And if you remember, chromatin is just DNA wound around histones. So chromosome is just like a really general term for just like where genes are stored. So it doesn't really give a whole lot of information away. However, one thing that is a really common point of confusion is that a chromosome looks like this, like an X, when a chromosome actually looks like this. It's just one. So here, down here on the right, you have a karyotype. And this karyotype is showing just a somatic cell from a XX individual. And you can see somatic cells are what we call diploid cells. Remember, diploid means that those cells have 2 of each chromosome. So for example, these cells would have 2 copies of chromosome 1. And so chromosomes are actually named based on, named in order based on size. So this would be a karyotype, and you can see like chromosome 1 is the largest, meaning it contains the most genes, and also just like visually is the biggest chromosome. Chromosome 2 is also large, but contains just a little fewer genes than chromosome 1, chromosome 3. But diploid, di means 2, and what this means is that these cells have 2 of each of these chromosomes. So this is a chromosome, and this is a chromosome. These chromosomes, this is essentially right here, is looking at the same thing up here. The reason these cells have 2 of each chromosome is because they're coming from both parents, right? So one of these chromosomes is coming from the father, paternal, and one of these chromosomes is coming from the mother. Or I guess I should say one chromosome is coming from sperm and one chromosome is coming from an egg. So you have a sperm and an egg, each having 23 chromosomes, and they come together and they form a cell that has 46. So 2 of each of those chromosomes. So we would say that this is the maternal and this is the paternal chromosome. In diploid cells, because these chromosomes, this is maternal and paternal, these chromosomes contain the same genes, but they're not identical. So what that means is, essentially, now you know this, these will have all the same genes, all the same loci, but these will be different alleles, or maybe different alleles. So for example, chromosome 1, if it has the gene for like eye color, you could have like the allele for blue eyes on this chromosome and the allele for brown eyes on this chromosome. It's an oversimplification. Eye color is like very, very, very complicated. But if we were to use that as an example, again, like one gene would be eye color, and then different versions would be like blue versus brown. So these chromosomes are the same. They're both chromosome 1. So they both contain the same genes. They both contain the gene for eye color, but they may be different versions of those. So maybe different alleles. So those are both chromosomes, right? So 2 copies of chromosome 1. So we call these homologous chromosomes. Homologous, homos, means the same, and logos or logi means kind of like, if I remember, it means like structure. So what that means is same structure, and it's referring to the fact that they contain the same genes. They're both chromosome 1, but they just have different sources, maternal versus paternal. So homologous chromosomes are just like the chromosomes that we have 2 copies of every chromosome in diploid cells. Haploid cells, which are your sperm and your egg, only have 1 copy of each chromosome. So haploid cells do not have homologous chromosomes. Okay. So we have homologous chromosomes. Again, this is looking at a karyotype in a cell. And this cell that this karyotype came from would have not gone through DNA replication yet. So remember, DNA replication, what that does, which is S phase of the cell cycle, DNA replication doubles the genetic information in a cell. And so what that means is you would go from this, which is 2 copies of each chromosome, homologous chromosomes, but DNA replication duplicates that, which means that after replication, you would have 2 copies of the maternal chromosome 1 and 2 copies of the paternal chromosome 1. So essentially, if you were to look at the same karyotype, like after S phase of the cell cycle, you would see a pair of chromosome 1. You would see 2 pairs of chromosome 1, is what I meant. So that's kind of like looking up right here. So this, again, these homologous chromosomes is basically what you're looking at here. So let's just say that this is homologous chromosomes of chromosome 1, right? So this is the paternal copy of chromosome 1. This is the maternal copy of chromosome 1. During DNA replication, we duplicate that. So we make a copy of all of the DNA. When we make a copy of the paternal chromosome, that means we have 2 copies of the paternal chromosome. We also, during DNA replication, make a copy of the maternal chromosome, which means we have 2 copies of the maternal chromosome. So DNA replication duplicated this chromosome, DNA replication duplicated this chromosome. When DNA replication happens and this chromosome is duplicated, those 2 chromosomes are kind of attached. And when those 2 chromosomes are attached, they are called sister chromatids. So sister chromatids are present after DNA replication. And this is very important. Sister chromatids are present after DNA replication, no matter what type of cell division you're doing, right? So this is the process of replication. This is what happens during replication, no matter what cell type you're talking about. This is what happens during DNA replication in somatic cells and in your pre-sex cells. So we'll talk about that in a minute. Well, or maybe on Monday. So DNA replication made this chromosome duplicate. And so we have sister chromatids of the paternal chromosome, and we have sister chromatids of the maternal chromosome. So remember, originally, these were called homologous chromosomes, the maternal and paternal. After replication, the paternal has sister chromatids and the maternal has sister chromatids. But now, all of these are sister chromatids are identical, right? So this is the paternal chromosome. These have the same genes, but also the same alleles, right? This is an identical, sister chromatids are identical copies. So these are two copies of identical chromosomes. These are also two copies of identical chromosomes. Together, these are what we refer to as a homologous pair of chromosomes. So a homologous chromosome are just a single copy maternal and paternal. Homologous pair is when we have done DNA replication, and so we have a pair of paternal chromosomes, sister chromatids, and we have a pair of maternal chromosomes, sister chromatids. So a homologous pair indicates that they're sister chromatids. So that means DNA replication has taken place. So homologous chromosomes are prior to DNA replication, sister chromatids, and thus homologous pairs of chromosomes are post DNA replication. So that's a little bit about chromosome terminology, and that is really important because as we talked about in the cell division lecture, what we're doing with cell division is separating chromosomes, right? So we want to divide DNA. With mitosis, we basically just want to go from like this and duplicate it, and then, you know, put one of these over here, 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 basically duplicate this. With meiosis, we don't want two identical cells, we want 4 cells. And so I think I have a summary slide. So this slide is just comparing mitosis to meiosis. And I think this is where a little bit of the confusion was, because the concept check asked, like, when are sister chromatids separated? And the answer was during mitosis, but also during meiosis too. So mitosis and meiosis both start with a parent cell that has gone through DNA replication. So what that means, so this is just looking at a cell that has like 2 chromosomes, right? So this would be like chromosome 1 and this is chromosome 2. So it's diploid. We have, like, you could say paternal and maternal, paternal and maternal. And each of those cells have gone through each of those chromosomes have gone through DNA replication. So you have sister chromatids of the paternal chromosomes and sister chromatids of the maternal chromosomes. Same for meiosis. The difference between mitosis and meiosis is that during mitosis, those homologous pairs, so remember, so these are homologous pairs of sister chromatids. The homologous pairs line up along the metaphase plate. So you can see like chromosome 1s line up on top of each other, chromosome 2s line up on top of each other. So what this allows to happen is that the sister chromatids are pulled apart during mitosis. So during mitosis, sister chromatids are separated, right? So you can see that, how those homologous pairs are lined up on top of each other. During meiosis 1, what happens is we still have the same chromosomes, but you can see something different happens after DNA replication. Those homologous pairs are actually kind of like joined together into what are called bivalents. And so that's just when you have those sister chromatids of the maternal chromosome and sister chromatids of the paternal chromosome kind of like stuck together. But this is really important because forming those bivalents is what allows those chromosomes to exchange genetic information via crossing over. We're out of time, but I just want to show you really quick. I don't want to hold you over, but I just want to show you when those bivalents, you can see the difference between mitosis and meiosis 1. Mitosis, those homologous pairs line up on top of each other, and so you can separate sister chromatids. During meiosis 1, the homologous pairs don't line up on top of each other, they line up next to each other. And so what that means is that instead of sister chromatids being separated during meiosis 1, homologous pairs are separated. And then it's during meiosis 2 where those sister chromatids are separated. But we will review this again next week. I also plan on reviewing a little bit about replication, so kind of clarifying Okazaki fragments and the difference between leading and lagging strands, all of that. But if you have any questions, you know, reach out, but we will talk about it. I have a whole lecture planned, and I love talking about mitosis and meiosis, which is surprising because I did not love it when I was in Bio 181, but I love it now. So happy to talk about it. Happy to talk about DNA replication. We are out of time today though. And so I definitely don't want to hold you over class, but I can hang out for a little bit longer. I am not planning on holding class on Wednesday because I want you to have freedom to enjoy a break or work on other stuff. However, I'm totally available. So if you would like to meet on Wednesday, you know, just send me an email. I'm happy to arrange something, no problem. And then I had a couple of questions. Let's see. On the Unit 5 checkpoint, the Unit 5 checkpoint is not technically your final because the Unit 5 checkpoint is basically just the same as like all the other checkpoints. It goes into your grade the same as all the others. Your final is technically the ePortfolio reflection questions. So that is technically your final, which is open, and you have until December 6th to work on it. Thank you so much. You're also wonderful. As for if there are any classes this Wednesday or if school is closed, let me look that up for you. I need to know that as well. Okay, school is not closed. Okay. Professor Chi-Chi, she's trying to mess with me. All right. So, okay, so there are classes on Wednesday. We are not planning on meeting on Wednesday, but I can if you want to. I'm happy to meet on Wednesday if you want to. But I didn't plan on it. So any other questions? I hope you all have just like a wonderful, wonderful break. I hope you have like a wonderful, like, I hope it is nice. If you are having a hard day on Thursday or don't have anybody to chat with on Thursday, let me know. You can Zoom into my Thanksgiving. I would love to have you at the table. We're making ham and turkey and lots of pie. So if you want to join Thanksgiving, you're more than welcome to join my Thanksgiving. I would love to have you there. Just send me an email. You're welcome. We would love to have you. I'd love to show you my parents, my daughter, my sister. We're loud and loud all the time. So we'd love to have you. Have a wonderful night. Hey.
Hi, Professor. I have a quick question about mitosis and meiosis. In the beginning, is there anything that's different between them before they start splitting up?
No, that's an excellent question. Oops, I forgot to share. So no, that is the thing about both mitosis and meiosis. Before they start splitting, like DNA replication is exactly the same. So the cells that are preparing for cell division are diploid. So they have 2 copies of each chromosome. They go through DNA replication to become quadruploid, where they have 4 copies of each chromosome, so homologous pairs. And if it's doing mitosis, the only difference between those cells will happen during the actual processes of cell division. So if a cell is going through mitosis, what will happen is the homologous pairs will line up along, if it's going through mitosis, the sister chromatids will line up along the metaphase plate. So that's super important, is that if it's going through mitosis, sister chromatids line up, because those are what are separated.
So like before DNA replication and before the synapses, like where crossing over occurs, is there anything different before that?
No, they're
So it's
exactly the same. So like interphase is the same.
okay.
That's a
Thank
good
you.
question. Yeah, that's an excellent question. That was actually like the reason for me, the thing I struggled with the most when I learned about mitosis and meiosis. I was like, what is the difference? But it's everything after interphase. Linda, please send me your completed recipe. You will get bonus points in the form of just like love. All right. Well, thanks everybody for being here. Happy holidays. Have a wonderful break. And again, if you need somewhere to go on Thanksgiving, we're keeping it pretty safe, but you're welcome to join me via Zoom. Love to have you. All right. I'll see you later. Wonderful break. Bye.